Welcome, doctors. Dr. Naveen here. So let us begin our topic, that is color vision. So what are the learning objectives of this webinar session? So I'll be talking about what is a Young-Helmholtz theory when it comes to color vision, and also I'll brief you regarding what is color blindness. So this is a short session on these two topics. And to begin with, when I say color vision is actually because of cones. As you already know, we got two kinds of photoreceptors inside the retina. One is rods and the other is cones. Rods are concerned with night vision, that is photopic vision, whereas cones are concerned with photopic vision and also color vision. So okay, so there are three primary colors, right? There are three primary colors, and one is red, other is green, and one is blue. So there are three primary colors when it comes to color vision. So what you need to understand is the cones have three different kinds of photopigments in them. When it comes to rod, I told you, right, rhodopsin is the photopigment, whereas cones are of three different types, each having a different kind of photopigment. One pigment is a blue sensitive, also known as short wave pigment. The second type of pigment is green sensitive or middle wave pigment, and the third variety is red sensitive or long wave pigment. So let me repeat it again. One is blue sensitive, other is green sensitive, and the third variety is red sensitive pigment. What is the difference? So when I say blue sensitive or short wave pigment, so this pigment absorbs light maximally in blue violet spectrum. So blue violet spectrum. So at this spectrum of light, the absorption is maximum. When it comes to green sensitive, it is during green portion, and when it comes to red, it is in yellow portion. Yellow, it also covers a segment of red, but to be more precise, it is yellow portion which is taken care by the red sensitive or long wave pigment. So three kinds of cones with three different kinds of pigments acting at different wavelengths, three different wavelengths. So this theory is known as trichromatic theory of color vision, also known as the one what we are discussing, that is the Young-Helmholtz theory. So it is simple now. What is Young-Helmholtz theory? It says there are three different kinds of cones with different kinds of photopigments, and these three different kinds, they help in appreciating three different kinds of colors, one with blue, other with green, and other with red. When I say color, there are three primary colors with different wavelengths. You know, for any color there is a complementary color. For any color there is a complementary color. What is this complementary color? Complementary color, when you mix it with its own complementary one, what happens is it produces a sensation of white. So mixing one color with its complementary color produces a sensation of white color. So that is what is complement color. And when it comes to color, there are three characters of color. One is hue, and the other is intensity of color, and the third variety is the saturation of color. So these are the three primary characters of a color, and these three are well appreciated by the cones. And so this is what is Young-Helmholtz theory. Let us move to the next one. So what is the chart you're seeing here? So this is Ishihara's chart. So what is this Ishihara charts? Ishihara's charts, they help in identifying color blinds. So what do? They help in identifying people with color blindness. So people with color blinds are identified by Ishihara chart. That is not the only test, right? There are few more other tests for detecting the color blindness. So what are the other tests which needs a mention? One is Farnsworth lantern test. This is second. And there is one more test known as HRR color test. So these are not important for exam, but one test which is very important you need to remember is Ishihara charts. They help in identifying a color blind. And what do they contain? They contain plates, color plates, which have figures made up of colored spots. You see, these are all colored spots, green and lighter green, red color. So they have a colored spots on a background of similarly shaped color spots. So when you identify the shapes are almost similar, but when you see the number has a different color in comparison to the background. So based on that, you can identify. If you are able to see this chart, it means you are not a color blinded person. But if you are facing any difficulty in reading the number which is mentioned there, so what is the number given there? You can you can answer that. That number is what I can perceive is 3. So if I am able to perceive it, it says that I am not color blinded. So this is how Ishihara charts, they help in identifying a color blind person. When I say color blindness, there are actually people can see 3 colors, 3 primary colors. What is that? Blue, green, red. So people who can perceive all the 3 colors are known as trichromats. So who are trichromats? Trichromats are the people who can see all the 3 colors. When I say dichromats, dichromats means they can perceive only one primary color. They can perceive only 2 primary colors. When it comes to monochromat, monochromat can perceive only single color variety. So this is different kinds of people who can perceive different colors. So now let us move to the genetics part of this color vision. Okay, so let me erase the text here. See, when I say color blindness, you need to remember one very important thing. Color blindness is X-linked recessive inheritance. But you need to understand one simple point here. Not all color blindness are inheritable in an X-linked fashion, because I told you, right, there are 3 primary colors. And when it comes to blue sensitive, what is blue sensitive? Otherwise known as shortwave color. The gene which codes these kind of cones is present on chromosome 7. And when it comes to green, that is middle wave, or red, that is long wave, these are located on Q, that is long arm of chromosome X. One statement is the most common form of color blindness is, that is red-green color blindness. When I say red-green color blindness, that is, it is X-linked recessive. The reason is these green-red cones which are helping in perceiving green and red color, they are located on chromosome X long arm. So that is the reason most of the times when I say color vision, it is transmitted in an X-linked recessive fashion. When I say that sentence, it goes without saying this color blindness is common in males. You know, in white population, 8% of the males are color blinded. When I say females, the females can also have a color blindness, but more commonly they have a blue color defect, because what happens here, blue is present on chromosome 7. So my statement doesn't infer that the green red color blindness is not present in females. It can also be present, and the phenomenon where X-linked recessive diseases also are seen in females is because of X inactivation. X inactivation can also cause green red or red green color blindness in females. The scientific term for it is lionization. What is that? Lionization is the scientific word for female getting an X-linked recessive disease. It should be usually seen in males. So this is a simple genetical overview about the color blindness part. And one more thing is, so you know that chromosome 7 is for blue-green. Chromosome 7 is for blue, and chromosome X, the long arm, is for green and red. Chromosome 3 is for rhodopsin. What is rhodopsin? Rhodopsin is a photopigment which is present in rods. And these two are for cones. I already told you one thing, right? Dichromats means they can perceive two colors, two cone pigments. Trichromats means they can perceive three cone pigments. Let us come back to the color blindness. When you want to name a color blindness, these are the terms which you use: proto, deutero, and tri. So what do you mean by proto, deutero, and tri? Protanopia means protanopia. What is prot? Prot refers to red. So red color blindness. Deuteranopia means green color blindness. And tritanopia means blue color blindness. So you need to remember that this is a memory part. Proto means red, deutero means green, and trito means blue color blindness. Among the three, there is a question you need to answer. Just look at the question. So my question is, heritable cause of color blindness with no sexual selectivity. So what is my question? Heritable cause of color blindness with no sexual selectivity. The options are: option A is protanopia, option B is deuteranopia, option C is tritanopia, and option D is achromatopsia. So what is your answer? You can type in the chat board there, and you can also type in the Insta, because this session is live streamed in Instagram also. So I'll give you 10 seconds for this. I'll be checking the answers here. The answer here is tritanopia. Why it is tritanopia? I already told you. Proto means red color blindness, deutero means green color blindness, trito means blue color blindness. The blue, the gene for blue sensitive cone is located on chromosome 7. Chromosome 7. So this is not a X-linked. That is, there is no sexual selectivity when it comes to tritanopia. So that is the answer. Then what is this achromatopsia? What is this new word? You know what happens when the light hits the retina? It is transmitted through the rods and cones and all the 10 layers of the retina. The final one is ganglion cell layer. The ganglion cell layer forms optic nerve, optic chiasma, lateral geniculate nucleus, and from the lateral geniculate nucleus, it is transmitted to, from the lateral geniculate nucleus, the light is perceived or transmitted onto the visual cortex, also known as the primary visual cortex. Where is primary visual cortex located? It is located around the calcarine fissure of occipital lobe. So around the calcarine fissure of occipital lobe, visual cortex is located, and this visual cortex has 8 areas. So in standard textbooks, they only gave 6 areas, but the latest Gannon mentions that there are 8 areas, and each area has got a different function. V1, the area V1 is associated with giving out the details of an image, that is the orientation, and also the edges of the image are processed in area V1. When it comes to area V2, image processing done on the larger visual fields, the bigger images, and V3 is concerned with the motion of the image. V4 area, the visual cortex V4 is concerned with the shape, color, and texture of the image. Before V8 came into picture, because of the researchers, V4 was believed to be the color center of the brain. So V4 is color center of the brain. V5 is also concerned with processing of the image when it comes to the direction of the movement, how it is going, like is it going to the left or right, up or down. So the direction is perceived with the help of area V5. When it comes to V6 helps in identifying the speed of body movement. So what is the velocity or speed of the image? And V7 is like visual field representation. The field is represented with the help of V7, the visual cortex. Not so important, but V8 is concerned with color vision. I'll see, here is the V8 located. Let us say a patient with a brain tumor is undergoing any surgery, or there is some head trauma injuring V8. What happens? The patient can no longer perceive colors, and the damage to area V8 can produce color blindness. and this pattern of color blindness is not anopia or nopia. This is called as achromatopsia. So what is achromatopsia? Achromatopsia is damage to area V8, maybe because of trauma, maybe because of iatrogenic surgical cause, leading to the color blindness is achromatopsia. So there is one more question here. The gene for blue sensitive shortwave cone is located on. So what is your answer? The gene for blue sensitive shortwave cone is located on. Take 10 seconds, 10-15 seconds of time. So answer it in the chat box. So what is that gene? As you know, the Q arm of chromosome is concerned with red and green. So the most common cause of blindness is red-green. It is located on X term. When it comes to chromosome 7 is not the answer. Chromosome 11 is not the answer. Chromosome 7 is concerned with blue. Okay. Chromosome 3 is concerned with coding rhodopsin. So this is the answer for this question is chromosome 7. So before concluding the session, I would like to give two more important points here. Number one, there is a drug known as sildenafil, otherwise known as Viagra. So it is used for erectile dysfunction. In cases of erectile dysfunction, this sildenafil drug is used. So what is the mechanism of action of sildenafil? The phosphodiesterase enzyme is inhibited, and as I told you when I am explaining you the phototransduction mechanism which is occurring in retina, phosphodiesterase enzyme plays a crucial role. Because of the inhibition of the phosphodiesterase enzyme by sildenafil, along with serving the purpose in erectile dysfunction, the major side effect or drawback of this sildenafil is it produces blue-green color weakness. So blue-green color weakness. And one important point is this is not a permanent, this is just a transient side effect, and once you stop the drug, it reverts back to normal because the phosphodiesterase is not irreversibly damaged. So the mechanism is there, it can be synthesized again and again. And last point before concluding the session is there any treatment for color blindness? No, there is either no cure or treatment for color blindness. We need to adapt to different teaching methods for color blinded people, and color blinded people has an issue in differentiating traffic lights. So during like most of the surgeons need to appreciate the red and green color structure, so it may be an issue for doctors too, and also the traffic heavy vehicle drivers, and there are some occupations in which the color blinded people are not allowed to work. So this will conclude our session. So let me give you a quick overview of what we learned today. So the session started with understanding the major function of cones, that is color vision, and also Young-Helmholtz theory, what is its importance, and the methods to detect color blinded people, and the genetics of color blindness, and areas of visual cortex and their different functions, and also two important like simple questions. Most of them are memory aspects, and there is a next webinar topic coming on 12th. So the topic is about Kawasaki disease, also known as mucocutaneous lymph node syndrome. So I'll be talking about the clinical presentation of a patient with Kawasaki disease and also the treatment aspects. So hope to see you there in that session. And you can log into www.rapidusmle.com and book your free tickets to join the event, and expecting your full participation. Thank you so much for attending this session.